Hello everyone, now we are going to discuss about the acute inflammation. Acute meaning sudden onset. So the onset in acute inflammation is from a few minutes to few hours. There are three major components of acute inflammation: vasodilation of small vessels, increased vascular permeability, and immigration or recruitment of the leukocytes. So these are the three major components: vasodilation, increased vascular permeability, and leukocyte recruitment or immigration of leukocytes. So now we will see the various reaction of blood vessels in acute inflammation. First is vasodilation. Mediator for this vasodilation is histamine. Due to vasodilation, there is increased blood flow or stasis of blood due to the vasodilation. The second reaction of blood vessel is increased vascular permeability. Increase vascular permeability. There are two mechanisms of increased vascular permeability. One is by the contraction of endothelial cells. As we know, the blood vessel is lined by endothelial cell. Whenever there is contraction of these endothelial cells, there is increase in the interendothelial gap and result into the increased vascular permeability. This mechanism is usually mediated by various cytokines or various mediator of inflammation like histamine, bradykinin, and leukotrienes. Another mechanism of increased vascular permeability is direct injury to the endothelial cells, which can be seen in thermal injury. Third reaction or response in blood vessel is responses of lymphatic vessels and lymph nodes. The fluid that accumulate in or produced during the inflammatory process is known as exudate, and this excessive exudate drain into the lymph node with the help of the lymphatics. This fluid carry leukocytes along with microbes, and this accumulate into the lymph node. As this is of infective etiology, this may result into the lymphadenitis or reactive hyperplasia. So this is usually noted in the inflammatory condition that adjacent lymph nodes are swollen or painful because of the drainage of this exudate into the adjacent lymph node. Now we will see the leukocyte recruitment to the site of inflammation. How leukocyte recruit or reach at the site of inflammation. The major leukocyte we will discuss or involved in the acute inflammation are neutrophil and macrophages. There are series of events before a leukocyte reach at the site of inflammation. First is margination. In the margination, normally blood cells flow inside the blood. Normally they flow in the midstream, but in inflammation they start marginating or start leukocyte toward the periphery of the blood vessel near the endothelium After this margination, they start rolling with the help of selectins. Selectins are the protein. There are three types of selectin: L-selectin present on leukocyte, E-selectin present on endothelial cells, and P-selectin present on the platelets. After this rolling, there is adhesion of leukocyte with the endothelium with the help of integrins. There are mainly two types of integrins, B-chem and I-chem. After the adhesion, these leukocytes transmigrate across the endothelium. This process is known as diapedesis. Diapedesis, that transmigration of leukocyte across the endothelial across the endothelium into the interstitium, and PCAM1 or CD31 will help in this process. Now once these cells or leukocytes in interstitium, now they reach to the site of injury or site of inflammation with the help of chemotactic signals. Chemotactic signals released by the various macrophages or microbes to attract the cells at the site of infection or inflammation. Now before going further, we need to understand or discuss about the selectins or integrins. The selectins, as we have seen, selectins are help in rolling process of inflammation. There are three types of selectin: L-selectin present on the leukocyte, E-selectin present on the endothelium cells, and P-selectin present on the platelets. Whenever there is a syndrome known as Cialel-Lewis syndrome or disease in which there is deficiency of selectins. Integrins are the proteins that help leukocyte to adhere with the endothelium. LFA1 integrins are present on neutrophil or monocytes. MAC1, VLA4, alpha 4, beta 7. These three integrins are present on the monocyte. So monocyte has all the integrins and neutrophil has only LFA1 integrin. As we have seen, the process of chemotaxis, when the site cells or microbes present at the site of injury attract the leukocyte in the interstitium, this process is known as chemotaxis, and there are some chemotactic agent, could be exogenous or endogenous. Exogenous chemotactic agents are bacterial products, for example N-formyl methionine, and endogenous chemotactic agents, mnemonic LIC used for them: L for leukotriene B4, I for interleukin 8, and C for C5a complement protein. This is a diagrammatic representation of the process of leukocyte recruitment. First of all, as we can see, this is the margination of the leukocytes from midstream to the periphery. Then we can see the selectins are over here. They help in the rolling process. After rolling, the leukocyte adhere with the help of integrin to the endothelium. This is the adhesion. After the adhesion, leukocyte transmigrate across the endothelium, and this process is known as diapedesis. Once the leukocyte is in interstitium, they follow the chemotactic signal released by the macrophages of the microbes and reach to the site of the infection or inflammation and result into the further phagocytosis of this microbes. Okay, now we will discuss about the phagocytosis and clearance of that offending agent. Two main types of phagocytes involved in acute inflammation are neutrophil and macrophages. So what are the steps involved in the phagocytosis? First of all, a phagocyte recognize the offending agent, attach with it recognition attachment of the particle to be ingested by the leukocyte. So first of all, phagocyte recognize an offending agent by various receptor. After this, there is an engulfment of this offending agent by the formation of phagocytic vacuole, and after this phagocytic vacuole formation, there is a killing of microbes by various enzymes. So what are opsonin? One important terminology is opsonin. What are opsonin? They basically increase the affinity for phagocytosis. They are coated microbes. They coat the microbes and increase their affinity for the phagocytosis. Examples of opsonin are IgG, C3V, mannose binding lectin, colectin. Leukocyte has various receptors for recognition of microbes or these degenerated particles to be phagocytosed, like G protein coupled receptor, toll-like receptor, cytokine receptor, and phagocytic receptor. After the recognition, they send a various signal and ultimately result into the inflammatory reaction or killing of the microbes. Now we will see about the phagocytosis and intracellular destruction of the microbes. As the phagocytic cells has receptor for the microbes, they recognize the microbes, then they start engulfment of the microbes and then form a phagosome. Phagosome has the microbe inside the vacuole. This phagosome fuse with the lysosome and form a phagolysosome. And in this phagolysosome there is degradation of the microbes with the help of lysosomal enzyme. How these lysosomal enzyme degrade these microbes? So in a neutrophil, there are a principal mechanism for degradation of these microbes or bactericidal system, H2O2 MPO halide system, which form a free radical of hydroxyl and chloride, which result into the destruction of bacteria or microbes by free radical injury. In macrophages, arginine is converted into the nitric oxide by inducible nitric oxide synthase and result into the formation of nitric oxide. Nitric oxide by combining with superoxide free radical form the peroxynitrate free radical, which causes free radical injury to the microbes. Now we will see about the various granules of neutrophils. As neutrophil is the principal leukocyte involved in acute inflammation, we will see the various granules of neutrophil. There are two main types of granules in neutrophil: primary or larger azurophilic granules, secondary or smaller specific granules. So primary azurophilic granules has MPO myeloperoxidase. They have bactericidal proteins like lysozyme, defensin, acid hydrolases. Neutral proteases like elastase, cathepsin G, proteinase 3, non-specific collagenase. On the other hand, secondary granules has lysosome, collagenase, gelatinase, lactoferrin, plasminogen activator, histaminase, and alkaline phosphatase. These various enzymes are responsible for the various function of neutrophil. Now we will discuss some of the enzymes and their function. Acid proteases, responsible for the degradation of bacteria and debris inside the phagolysosome. Neutral proteases, they are responsible for the degradation of extracellular components like collagen, fibrin, elastin, etc., and they are responsible for the tissue destruction. Neutral proteases also cleave C3 and C5 and they release kinin. Neutrophil elastase is another enzyme which is responsible for the degradation of the virulence factor of bacteria. So this is very important in controlling the inflammation or infectivity of bacteria. There are some anti-proteases in the body. They control the harmful effect of proteases. For example, alpha-1 antitrypsin, alpha-2 macroglobulin. Alpha-1 antitrypsin is very famous. Alpha-1 antitrypsin inhibits neutrophil elastase, and in case of deficiency of alpha-1 antitrypsin deficiency, there is sustained action of neutrophil elastase and will result into the destruction of elastic fiber support, and this is seen in one of the very famous disease of emphysema in lung. And now we will see one more important topic related to this: neutrophil extracellular traps, NET. So in infection or inflammatory mediators, they stimulate neutrophils to form extracellular traps which consist of viscous meshwork of nuclear chromatin and concentrate granules protein. Viscous meshwork of nuclear chromatin and concentrate granule proteins, they make the extracellular trap, and this net basically trap the microbes and kill them. Net formation is via chromatin decondensation. So arginine deaminase convert arginine into the citrulline, and MPO elastase then enter into the nucleus and causes the further decondensation of the chromatin, and this will help into releasing of this chromatin outside the cell and trap the microbes. Next, when a neutrophilic extracellular trap seen in SLE, systemic autoimmune disease, in which this is a source of nuclear antigen. Leukocyte-mediated tissue injury. If there is an inflammation or leukocytes recruit over there, then they will cause collateral damage to the adjacent tissue. They might cause an autoimmune disease or host react excessively, which is seen in hypersensitivity reaction. So this is the difference between neutrophil and macrophages, which we have already discussed in one of our video. There are certain anti-inflammatory cytokine which actually prevent or suppress inflammation, TGF beta and interleukin 10. So thank you for listening. In the next lecture we will discuss about the mediators of inflammation.